Welcome everyone. My name is Yiwei Li, currently a PhD student at the University of Western Ontario. Today I'm presenting our recent research, DELPHI, an accurate deep ensemble model for protein interaction sites prediction. This is a collaborative work between two Canadian universities. The co-authors are Brian Golding from McMaster University and Professor Yiwei Li from University of Western Ontario. Here's an overview of today's talk. We will start with some background information and define what the problem is. Next, we're going to introduce some previous methods, including the commonly used classifiers and the features. Then we'll move on to the introduction of DELPHI. We will focus on the model architecture, the datasets, and the features. And last, we will show some performance evaluation results, as well as some biological insights. So let's get started with some background information. Proteins are essential to organisms, and they participate in pretty much every process within cells. It is estimated that the human body has about 1 to 3 billion proteins, so proteins are very important. The function of proteins vary, such as being antibodies, enzymes, hormonal proteins, structural proteins, storage proteins, transport proteins, etc. In general, proteins are made of 20 kinds of amino acids. The primary structure of a protein is its amino acid sequence. Proteins also have secondary, tertiary, and quaternary structures, and these are just fancy words of saying 1, 2, 3, 4. Protein secondary structure represents local structures within the protein itself. The most common structures are alpha helix and beta pleated sheet. The tertiary structure contains the overall shape of a protein molecule. In other words, the spatial relationship of the secondary structure of multiple protein chains. The quaternary structure describes the structure among several protein molecules, which is a protein complex. The protein sequence are the most available data for proteins, and our work focuses on predicting from the primary structure only. Proteins are the most important molecules in cells. They carry out most of the cellular processes, and quite often, they keep the cell functioning by interacting with other proteins. This process is called protein-protein interaction, or in short, PPI. This figure is an illustration of two proteins interacting. Identifying PPI information helps improve the system-level understanding of molecular processes. The bonding amino acid residues are protein-protein interaction binding sites. If looking at this picture, the contacting amino acids from the blue and the purple proteins are the binding sites. Detecting PPI binding sites is also a hot topic. It helps understanding cell regulatory mechanisms, locating drug targets, predicting protein functions, etc. So let's define the task of DELPHI here. DELPHI is a program that predicts protein-protein interaction binding sites. The input of DELPHI is the primary structure of a protein, which is basically the sequence. The output of DELPHI is the binding sites of that protein. Showing this in the screenshot at the bottom of the slide, the first two lines are the input protein name and the sequence in FASTA format. The third line is the prediction results of DELPHI on each amino acid, where 1 means binding sites and 0 means non-binding sites. Now that we know the definition of the problem, so let's take a look at some of the previous methods. There are some experimental methods where you can determine the binding sites in wet labs. The description of these methods is beyond the scope of this presentation, but usually they are labor and time intensive, and that's why many computational methods have been proposed. The advantage of computational methods is that they're high throughput, which means it can predict many binding sites in a short period of time. Prediction problems in bioinformatics are usually converted into classification problems in computer science. In such a problem, you have two phases: training and inference. The first, you train your program or models with existing knowledge, and then when there's a new input, the trained program classifies it into different categories. To perform such tasks, a classifier is needed. Very few programs for binding sites prediction are algorithmic based, for instance, sequence similarity based or alignment based. All others are machine learning based. I'm showing a few machine learning classifiers that have been used in this problem. For example, support vector machine, logistic regression, Bayes classifier, random forest, and recently in deep learning, CNN and RNN. We know that one of the most important steps in developing machine learning models is to represent the input with certain features. I'm listing a few widely used features in this problem. The most effective feature is probably the position-specific scoring matrix, or PSSM. It is computed by using BLAST, where multiple alignment is performed on the input sequence against a BLAST database. This extracts a lot of information about a protein. There are also other features describing certain properties of amino acids. For example, solvent accessibility, relative amino acid propensity, hydropathy index, physiochemical characteristics, and recently inspired by natural language processing, protein embeddings have also been explored, where each protein is treated as a sentence and each chunk of the sequence is treated as a word and is represented by a vector. So, followed by what people have done in the past, we developed a new deep learning model called DELPHI. Our goal is to have a model that has better performance in terms of accuracy. So let's get into it. We use three sets of data to train and evaluate the performance of DELPHI: training, validation, and testing. The model is trained on the training data and validated on the validation data to avoid overfitting. After several iterations of tuning, the model is obtained and ran on the independent testing data, and then the performance on the testing data is reported. Notice that the model never observes the testing dataset during the training and the validation process. We choose firstly the testing datasets, then construct the training datasets such that they are not similar with testing. The testing datasets were previously constructed from other publications. Dataset 448 was built in 2019. Dataset 355 is a subset of 448 because the training set of one of the state-of-the-art programs, DLPRED, has a big overlap with it. So we removed those proteins and left with 355 proteins. Dataset 164 was built in 2014, and dataset 186 and dataset 72 were constructed in 2010. We construct next our training and validation data such that there's no similarities between training and validation. There's also no similarity between testing and the union of the training and validation. DELPHI has a relatively large training data because this is beneficial for deep learning models. The details of these datasets can be seen from the DELPHI paper, and the slides shows some statistics of them. All datasets are available to download on DELPHI's website. As shown in this slide, we use 12 features in DELPHI to represent a protein sequence. The first column is the feature names. The second column indicates whether the feature is computed using other programs or loaded as values directly. The third column is the dimension of each feature. Among the 12 used features, the first 3 are new, and the rest have been used in previous publications. The first new feature we use here is high-scoring segment pair, or HSP. An HSP is a pair of similar subsequences between two proteins. DELPHI will add a score to testing subsequence if it's similar with a subsequence in the training data, which also contains binding site. The second new feature we use is ProtVec 1D. We developed this feature based on ProtVec, which was published in 2015. ProtVec uses Word2Vec to construct a 100-dimensional vector for each 3mer. We collapse the dimension by making the summation of the 100-dimension vector, and we have experimentally verified this renders very similar results as the original embedding for this problem. And this way it uses much less memory comparing to the original embedding. The third new feature is position information. This position feature is inspired by networks like BERT, where the first position gives the value 1, second position gives the value 2, and so on and so forth. All features are normalized to a value between 0 to 1 before feeding to DELPHI. Now let's look at the input and output of DELPHI model. For each input sequence, a sliding window is applied on top of it. The window is of size 31, and we shift one position at a time. For each window, DELPHI predicts the binding probability of the centered amino acid based on the information of all the 31 neighbors. At the end, each residue in the input sequence will have one predicted value. Now, this slide shows the architecture of the DELPHI model. Each input sequence is firstly represented using the 12 features we just introduced, and then passed to the DELPHI model for inference. DELPHI has an ensemble architecture with two components, one CNN and one RNN. The intuition is that different components can learn different things from the input features, and combined together, they do a better job. The two components are trained separately, as shown on the left and the middle picture. And we use a fine-tuning process. The CNN model on the left has a convolutional layer, a max pooling layer, and fully connected layers with dropout. The RNN component in the middle has a bidirectional GRU layer and fully connected layers with also dropout. After tuning the CNN and the RNN model, we freeze the weights of the core layers, which are the convolutional layer and the bidirectional GRU layer. And then we move on to the right picture, which is the final ensemble model. We load and freeze the weights of the convolutional layer and the bidirectional GRU layer that we have tuned previously, and then we train only the fully connected layer. And that's the process of how we obtained the DELPHI model. Now that we know how DELPHI works, so let's take a look at some of the results. The first performance comparison here is on dataset 448 and 355. We comparatively evaluated DELPHI with other 9 state-of-the-art programs. We have computed 8 different evaluation metrics: sensitivity, specificity, precision, accuracy, F1 score, MCC, area under ROC curve, and area under PR curve. Since binding site prediction is an imbalanced problem, which means the number of non-binding sites is much more than the number of binding sites. So MCC and area under PR curve will be a better indication of the overall performance. All numbers here are shown using heat maps. The darker the color, the better the performance. As we can see here, DELPHI has the highest performance in all metrics. It surpassed the second best program in MCC and AUPR by large margin. To further compare DELPHI with other programs, we used another 3 older datasets. And as you can see here, again, DELPHI achieved the best performance in all metrics. We did an experiment to show that all 12 features are useful. We removed one feature at a time, and the remaining 11 features are used to train and then evaluate the DELPHI model. As shown in this figure, the leftmost bar is using all 12 features, and the performance decreases with the removal of any feature. This shows that all features play a part in the prediction. It is probably expected that removing PSSM creates the biggest performance gap. But our newly introduced features HSP, ProtVec 1D, and the position information are also very useful. We did another experiment to show that the Ensemble Architecture and the three novel features improved the performance. We evaluated the CNN, RN, and the Ensemble models separately, with and without the three newly added features. So in total, 6 tests. We plotted the value of area under PR curve and MCC of each test. And clearly the Ensemble model outperforms the individual CN and RN model. Also, the improvement due to the 3 new features is even higher. We did another experiment to show the correlation between DELPHI predicted scores and the level of conservation. We evaluated 3 proteins. They are the transcription factor SH2D2A, the alpha subunit of the hemoglobin protein, and the SRY protein. The blue dots are the computed conservation scores for each position, and the green dots and yellow bars are the mean and standard deviation of DELPHI predicted scores. Figure A and Figure C show that the locations with high DELPHI scores are also the sites with high degree of conservation. As expected, this correlation is not perfect because sequence conservation can occur for many other reasons other than protein-protein interaction. The SRY protein in Figure B binds to sites in DNA rather than proteins. This is the reason for high conservation around position 190 to 260. DELPHI score is floating around low values except 2 sites. We have run DELPHI on the entire human proteome, which is about 20,000 proteins. The predicted results are available on the DELPHI website. The trained model, source code, and data processing pipelines are open sourced on GitHub. DELPHI is also available as a web server. We hope this makes the job easier for users with less computer science background. All datasets and test results we just showed are also available on DELPHI's website. We welcome you to use DELPHI, and thank you very much.